Welcome all to discuss a Module 5, Stages of Formulation Development, Part 1. In the previous module, we have discussed in detail about a quality-based design and its components. Now, we have come a stage where the actual formulation development activity happens with respect to dosage form. Here, we have described taken certain formulation, a dosage form, and certain examples and certain challenges, what all factors are involved for the particular dosage form. First of all, any formulation we develop, the sourcing of active pharmaceutical ingredients are very important, because if there is no active pharmaceutical ingredients and there is no product. For example, phenydiene sodium, paracetamol, pemetrexed, and etc. There are a lot of drug substances you must have studied in pharmacology and as well as the medicinal chemistry. So these all the components are called as active pharmaceutical ingredients, which gives the therapeutic activity. So sourcing of AP is one of the critical tasks for the development. There are multiple factors based on dosage requirements. Here we have tabulated a few factors for your understanding. For example, a critical factor is to be considered for each dosage form. For the tablets, apart from the regular test, you may have to consider a particle size, bulk density, tab density, and solubility. We understand that what is the importance of particle size and bulk and tab density for any kind of tablets and capsules. Similarly, for the parenterals, you need a solubility, microbial attributes, apart from your regular test parameter, which is normally your assay of drug content, degradations, related components, residual solvents. These are the routine test parameters which commonly being followed for pharmaceutical active ingredient. And topicals also needs certain controls on particle size and microbial attributes, because these are the factors required for the respective dosage forms. A selection of drug substance. What importantly we have to look at when you select a drug substance, which is nothing but active pharmaceutical ingredient and an excipients. Always in Indian pharmaceutical industry, they select the API or excipients based on their quality of the services and quality of their regulatory norms, and as well as they call it, which is DMF, which is nothing but drug master file. Whenever we develop any formulations for ANDA or NDA, the DMF is important file needed for any of the active pharmaceutical ingredients. A formulator should ensure that the DMF should be in place for any active pharmaceutical ingredient. Apart from that, when you work on an excipients, you may need to assess other tests, particularly on microbial testing, heavy metals, and if you are working with the rubber stoppers, you may need to check the leachable and extractable for the rubber components also. The supplier maybe provide all these informations on your request, and you can get assessed based on your experience, and that you need to be compared against at least two to three different a manufacturer or the supplier. Here we have just put it for your reference APA-COA of paracetamol and the material safety data, which is something like which is needed for any components. If you handle it, you should understand what is a safety data sheet, which can provide a lot of information to handle those substances in our laboratory. A typical sample APA-COA of paracetamol are presented here. You can see that the specification and the results of the component. The test parameters have been tabulated, which includes appearance, solubility, identification, melting point. All these parameters are very important. Believe this component is being used for the parental because you could see these components are controlled with microbial, particularly on aerobic, mold and yeast, and endotoxin also. So it is believed this APA is being used to prepare a parenteral formulation. MSDS for the sodium chloride, a technical data sheet for trisodium citrate dihydrate, and CFA of the sodium chloride. These are the main factors or the attributes which formulation scientists have to look at before they start the product development. A specification should be defined what kind of input material is needed for me to formulate a product to be filed for an ANDA or NDA, which is nothing but you can go to a global filing also. MSDS typically gives us all the physicochemical characteristics and its ignition temperatures, and as well as the disposal procedures and the safety and stability of the components is clearly defined for most of the excipients. You can avail these information from the respective manufacturer and assess how safe is the component to handle it at your facility. What are the criteria for the selection of an excipient? As we discussed well, for the parental dosage form, influence of excipient on the overall quality, stability, and effectiveness of drug product. And then a compatibility of excipients with the drug and the packaging system. Any excipients you use it should be compatible with your drug and your components. It is a vial, rubber stoppers, or an ampule. Any other primary packaging material should be compatible. Either you have to select a right grade of an packaging system or it should be a excipients also. In both the way, we need to get a top grade material to be used, and the specification also should be highly controlled as per the drug substance or excipients material attributes. And also there are certain cases you could see the preservatives may be absorbed through the certain tubes and the rubber stoppers and the filters. It is very high recommendation should be provided to the manufacturer when we select these components. And also during the lyophilization process, there are certain acetate buffers will be lost. You need to investigate and understand what is the process is being involved when this acetate buffer losing is the buffering system. And also the amount of percentage of an excipients used into the formulations to be assessed in terms of the regulatory. Normally in FDA, you can get this information through inactive ingredients database, which provides you a dosage and administrations and as well as the percentage of amount is being used So that would be the backup data for you to use those amount for your formulation. Route of administration. That is also is one of the criteria, because there are certain drugs or certain excipients, preservatives should not be used whenever in contact with brain tissues or CSF, particularly the route of administration, intracisternal, epidural, intradural injections should be preservative free. Also, it is preferred that the drug product to be administered via intravenous route be free of particulate matter. However, if the size of the particle is well controlled, like in fat emulsion or colloidal albumin or amphotericin B dispersion, can be administered via IV infusion. Again, the dose volume, because we know that there is a difference in LVP and SPP. A dose, if it more than 15 ml required by the EP to be preservative free unless it is justified. Normally the LVP they call it, if it is more than 100 ml, it is called large volume parenteral. If it is less than 100 ml, shall be called as small volume parenterals. The USP recommends that special care be observed in the choice and the use of added substances in preparations for injections that are administered in volume exceeding 5 ml. Whether the product is intended for a single or multiple dose. According to USP, the single dose injection should be free of preservative until it is justified. The FDA takes the position that even though a single dose injections may have to be aseptically processed, the manufacturer should not use a preservative to prevent microbial growth. But however, it is case to case. There are certain products comes with the market, even if it is a single dose, the product comes with preservative. This has to be assessed and approved from an FDA before we initiate the formulations. European agencies have taken a more lenient attitude on this subject. The length or duration of a time that the drug product will be used once the multi-dose injection is opened. A multi-dose most of the time can be used 7 days or 14 days or 28 days, or in sometimes 3 times a day. So these all, the multi-dose comes with certain level of preservative which could be studied as part of development. We call it as in-use stability. The in-use stability shows that how a drug product should be used and what is the level of a preservative should be used and what is the efficacy when you add such amount of preservative into the formulation. A detailed study and in-use study should be performed as a part of development. Again, the regulatory perspective. The excipients included in the GRAS list does not mean that the excipients can be used in the injectable. The excipients which are meant for IVIM, SC or IP, etc. need to be mentioned in GRAS list, and then only the excipients qualifies to be used for parenteral dosage form. The United States and Europe require all excipients to be developed, declared along with their quantity on the label. What is put on the immediate container if the product is in injectable preparations. And also, as we have discussed, inactive ingredients database gives you a sufficient information for you to select the right amount of an excipients when used for your drug product formulation. In Japan, only the excipient names are required in the labeling information that is included in the product, such as pack insert. In EU, all the excipients must be declared under labeling if it is a medicinal product is an injectable, a topical or an eye preparation. The European guide for the label and package leaflet also lists excipients with special issues and are addressed. A package leaflet must include a list of information on those excipients knowledge of which is important for the safe and effective use of medicinal product. Similarly, 21 CFR 201.22 requires a prescription drugs containing sulfides to be labeled with a warning statement about possible hypersensitivity. According to the notes for the guidance on pharmaceutical development, the choice of excipients, their grade, compatibility, concentration, and function should be described in the P2 sections of the Common Technical Document. Common Technical Document which has to be prepared by the drug product manufacturer and which has to be updated or informed to the agency how these excipients have been selected and its performance. It is necessary to justify inclusion of all ingredients in the drug product and describe their intended functions. A specification of plus or minus 10% at the end of shelf life is acceptable except for an antioxidants and preservatives, where performance data from PET or stability data may justify broader limits. Bioburden and endotoxin limits of excipients used in the manufacture of sterile medicinal products shall be stated. Individual testing of excipients may be omitted. If an excipient is present in PHUR or other major pharmacopoeia, the monograph specifications are usually acceptable in the registration file. For excipients not described in any pharmacopoeia, specification should include physical characterization, identification test, purity test, assay, and impurity test. A certification must be included to confirm that excipients are of non-animal origin. This something call it a TAC or BSc. Certifications should be obtained from a vendor. Most of the products, even you can refer the excipients or the API, the list of drugs, the list of tests have been provided in the pharmacopoeia. You can refer those tests and request a minimum test should be complies as that of the respective pharmacopoeia. If this is not the case, a regulatory agency will require a documentation to demonstrate a freedom from the viral and transmissible spongiform encephalopathies, which is nothing but TSE, and bovine serum encephalopathy, which is nothing but BAC risk. European Commission directive requires manufacturers to provide a certificate of suitability or the underlying scientific information to attest that their pharmaceuticals are free of TSEs. Excipients for label and corresponding information for the pack insert. For example, here we have just provided certain examples for your better understanding. Benzoic acid and benzoyase, the threshold level is 0. The risk of jaundice in newborn babies. These information has to go in your package leaflet whenever you are using these components into your formulation. Also, ethanol and propylene glycol respectively, the labels should be defined. The medicinal product contains small amounts of ethanol, less than 100 mg per dose. For a propylene glycol, may cause alcohol-like symptoms. If ethanol is used between 100 mg to 3 gram per dose, the medicinal product contains volume percentage of ethanol that is up to mg per dose equivalent to ml. A beer dash ml a wine per dose. Harmful for those suffering from alcoholism, to be taken into account in pregnant or breastfeeding women, children and high-risk groups such as patients with liver disease or epilepsy. Challenges with the drug substance and excipient selection. Generally, excipients and vendor selections can greatly influence development time, performance, quality, and acceptance of final products. Consequently, quality excipients suppliers should maintain a DMF, which is nothing but drug master file, with FDA for non-commercial items. Consistently conform to monograph requirements and manufacture an ISO 9000 certified facility. And also the facility should have at least FDA inspected and audited by either pharmaceutical companies or International Pharmaceutical Excipients Audit. These are the very basic necessary informations to be gathered whenever you start developing a formulation for submitting to ANDA or NDA. So we have discussed with detailed about selection of API and excipients in detail. Now coming to the packaging material. In pharmaceutical industry, we have to give an high importance when we select a sourcing or when you select a packaging material, particularly on the primary. You have to give high equivalency as that of your excipients and as well as active pharmaceutical ingredients, because these all components are getting interact with your drug product and gives you the stability based on the quality of the packaging material. Packaging material decides our final product's physical and chemical characteristics, protective needs and marketing requirements. There are multiple packaging materials are available with respect to dosage form. For example, a tablets capsules require a bottle pack or blister pack, aluminum alu blister pack, and in injectable need a vials, a glass vial, plastic syringes and glass pre-filled syringes and ampoules. And a dermatological, the cream requires a tubes, applicators. There are many other packaging materials are available based on the dosage form. A desirable attributes that packaging material must have as per FDA, the compatibility and safety concerns raised by the route of administration of the drug product and the nature of dosage form, example solid or liquid based. Kinds of protection the container closure system should provide to the dosage form, example photosensitivity, hygroscopic, easily oxidized drug product. Potential effects of any treatment or handling that may be unique to the drug product in the packaging system. Patient compliance to the treatment and ease of drug administration. Of course, the safety, efficacy, and quality of drug product throughout its life. Any material, when you select it, it should be compatible with your dosage form, because there are a lot of degradations happens from your primary packaging material. Because, for example, if you select a glass vial and the rubber stoppers for oxygen sensitive product, it is very important that like the size of packaging materials are very important, because if you keep it a more size, then you may need a more head space to be refilled with your inert gas, which is inefficient process. And also certain easily oxidized product need to be controlled with a suitable packaging system, and hygroscopic materials should be packed at least primary and secondary with certain level of inert gas. And there are certain potential components to be handled in a proper way by packaging and providing a certain labeling on the primary pack container with necessary precautions. The classification of packaging material. A packaging material can be classified into three terms, which is primary, secondary, tertiary, which is nothing but a primary packaging when a product directly interact with a drug product. The secondary is above the primary pack and the tertiary is above the secondary pack. It's a simple terminology. So we all understand that the glass vials and rubber stoppers is a primary pack for parenteral solutions, nasal drops, and certain level of ophthalmic solutions. A bottle, a blisters, a liners shall be a primary pack for tablets and capsules. So types of packaging material, what we have discussed: glass, plastics, metal, and rubber. Here we have just listed out what all the types of packaging material is being used in the formulations: ampoules, cartridges, syringe, tube, bottles, and vials, and blister and strip pack, stoppers, bottles, tubes, sachets, pumps, containers, and closures for the tablets and capsules. The tubes also certain times and blister and strip pack are being used for dermatological products. Of course, certain level, the parental formulations need certain flip-off seals and stoppers also. So these all components are made with rubbers, and metal is being used to make tubes, a strip pack, and flip-off seals. Of course, the plastics are all the sachets, bottles, pumps, container closures. And the glass is being used to manufacture ampoules, vials, syringes, and etc. Packaging materials depending on dosage form packaging. For example, a critical factors to be considered for tablets: bottle pack and blister pack. For an ophthalmic, glass vial, ampoules, glass and plastic prefill syringes, plastic bags, plastic bottles also. And topicals, tubes and bottles. Glass containers is being categorized in different types based on the hydrolytic resistance and the composition what is being into the glass composition. For example, a type 1 which comes with a borosilicate glass, which is being considered for any parenteral preparations, basically for acid, neutral, alkaline pH, and it has a good thermal shocks and the resistance. And similarly, type 2, a soda lime glass with a suitable treatment of inner surface to increase the hydraulic resistance. At certain level, these components can be used for parenteral preparations. Either before and after filling, you can do a sterilization. And type 3 is soda lime silica glass, which normally is recommended for non-parenteral preparation because it has average hydrolytic resistance. And type 4 is completely to be used for non-parenteral formulations. It is being used for a general purpose, not for any kind of administrations. There are few challenges when you develop a glass packaging components. Here we have just listed out few problems majoritarily found in pharmaceutical industry. Stability and efficacy during high temperature and stress conditions. Because glass is a container which can conduct the heat quickly to the formulations. Based on that, we will have a certain issue on stability of the product. And also the efficacy also can be down when the stability goes down. Problem 2 is particulate matter and delaminations. Many cases, if you see, if the degradation happens or the glass get erode due to a delamination process, the glass surfaces leach out into the bulk solutions and shows certain kind of particulate matter on storage. This is a very critical fact when we do a parenteral applications. And also thermal stability and customized drug delivery systems and product strength. The product strength also it matters when you use a glass surfaces because based on the strength, the components, the glass components or the rubber components may get leach out into the formulations. And whenever you change the packaging component, the customized one would lead a lot of challenges because the each area like kind of curves and bottoms which is more prone for leachables. So these are the very basic informations. Maybe you might have gone through a lot of information during your course. Here we just listed out what are the major criteria you have to consider while doing a formulation development. Coming to plastic container. Plastic container consisting chiefly of one or more organic components of high molecular weight which can be molded into desired shapes and hardened for use when subject to heat or pressure with or without the addition of some additive. They are used for many different types of packs includes ridged bottles which serve as packaging system for solid dosage form, sterile plastic packaging systems for human blood and blood components, plastic packaging systems for aqueous solutions, bags for parenteral solution and etc. There are lists which keep going. It is always his responsibility of a formulation scientist should understand on the packaging systems which is being used for the respective dosage form and what kind of dosage form you are selecting and what kind of a packaging material you are using based on the stability of the component. The types of plastic packaging systems into divided into two categories, which is thermoplastics and thermosets. We all know that the thermoplastic, which thermosoftening plastics, which can stretch based on the conditions, on thermal conditions, and these components mainly used for infusion bags and certain kind of dermatological products. For example, it is polyolefin, polyvinyl chloride, and polypropylene. These all components are called thermoplastics. Thermosets, which is highly stable with respect to a temperature, where these all shall be used for a more stable products. Here we have just described what is thermoplastics. These are heat softening materials which are usually rigid at operating temperature but can be remelted and remolded when exposed to high temperatures and pressure. When frozen, however, thermoplastics become glass-like and subject to fracture. Examples: polyvinyl chloride, polystyrene, polypropylenes, polyethylenes, and polyesters. Others maybe, the list keep going. The thermosets, they are called thermosets because they get distinctly infusible or insoluble when exposed to high temperature or heat and thus cannot be remelted and remolded after their initial heat forming. They are produced by polymerization process involving a curing or vulcanization stage during which the materials become set to a permanent state by heat and pressure. At drug plastic considerations, there are few parameters which you have to consider because there are certain cases few parameters should be considered based on the dosage. For example, permeation. The transmission of gases, vapors, or liquid through plastic packaging materials can have an adverse effect on shelf life of drug. Permeation of water vapor and oxygen through the plastic wall into the drug can present a problem if the dosage form is sensitive to hydrolysis and oxidation. Temperature and humidity are important factors influencing the permeability of oxygen and water through plastics. An increase in the temperature increases the permeability of gases. Leaching. There are many components. Over a period of time, the chemical components get leached based on the interactions with the drug products. Either it is a solution or solid substances. Those chemical components from the plastics which get entered into the drug product, and it shows certain kind of degradations or drug contaminations. This also is one of the major factor while you select a packaging material, what kind of a product you are using, what is your extractable and leachable of the particular component. Sorptions. It may be defined as bonding of a solute to a plastic. This process involves the removal of constituents from the drug product by the packing material. Sorptions may lead to serious problems for drug permeations in which important ingredients are in solution, since the drug substances of high potency are administered in small doses. Losses due to sorptions may significantly affect therapeutic efficacy of the preparation. Chemical reactivity. Certain ingredients that are used in plastic formulations may react chemically with one or more components of a drug product. At times, ingredients in the formulations may react with the plastic. Even micro quantities of chemically incompatible substance can alter the appearance of the plastic or the drug product. With this, we cover module 5. We have tried to cover a formulation development with certain examples and what are the importance of active pharmaceutical ingredient, APIs and packaging material, and what are the challenges while selecting the packaging materials are discussed. Moving forward, in the next module, we can discuss in detail in other formulation factors are involved during the product development. Wish you good luck.